Good evening, children. Good evening, everybody. How are you? How are you? Good? Yes. Tired? Back from school? Okay, I can see Shillong logged in. How are you, Shillong? How was your day at school? Yes. How was it? Yes, Shillong. Tell me, how was your day at school? Okay, I'm not able to see you people. Camera port number is 8084, isn't it? Okay. Okay, shall we start our class? Any doubts from the last class? Yes, children, any doubts? Shillong? Any doubts from the last class? No, yes? Nothing? No doubts? Okay. Yes, Shillong? Children, no doubts? So what were we discussing in the last class? The last thing what we talked about in last class, what was it? And how was your test? So many questions at a single time. So how was your test? First thing. How was it? Yes, children. How was your test? Yes. So we'll do one thing. We'll start with our chapter. In the meantime, we'll talk about your test and everything. So this was a chapter we were talking about excretory waste. And its removal. Okay. So what first and foremost thing, what a type of excretory products we talked about in the last class? Yes. What are types of excretory products? I divided it into 3 categories. What was that? It was ammonia, it was urea, and it was uric acid. Isn't it? These were 3 types of products we thought of in the last class. Okay, let's see one by one the characteristics of each product. Just a brief revision. Ammonia was found to be most toxic. This was moderate in toxicity. And this was what? This was least toxic. So if it is most toxic, it required a huge volume of water. Isn't it? This required a moderate volume of water. And this was insoluble in water. Isn't it? So I told you that day also it forms precipitate in water. Okay? It's clear? This was clear, I guess. Isn't it? Yes. No responses till now, Shillong? Yes, Shillong? Yes, children? Answer. Good evening. Good evening, Shillong. How are you, children? Good? This was clear, isn't it? This was what we were talking about in the last class. Just give me a thumbs up if it was clear to you in the last class. Everybody, just give me a thumbs up if this was clear to you. Everybody. Okay. Yes, Shillong. Thumbs up, please. Clear? This is 11th biology. Just wave your hands. Just to cross check. 11th biology, just wave your hands, children. Yes, this is 11th biology. So it was clear? Was it clear in the last class? Okay, so I'm proceeding. So I guess we did with the structure of kidney. I remember. We did with the ultrastructure of kidney where I told you the whole structure of the kidney. We talked about cortex, we talked about medulla, we talked about medullary pyramids, and the medullary pyramids have this structural and functional unit called as nephron. So today we are going to continue with the structure of nephron. Isn't it? Am I correct? This was the thing we are supposedly going to do today. So we'll start up with structure of nephron. So structure of nephron, first and foremost, we have a little bit of idea, isn't it? We have done these things in our preliminary classes in our junior school. So the same structure will remain now also, but small detailing. Obviously, if you grow up, you have to have some explained work also. Just keep on drawing with me, everybody. Oh. Structure of nephron. Let's label it. First complete this whole structure, then we will start labeling. Okay, this is a well labeled diagram of kidney. Okay, now efferent arteriole, efferent arteriole, I mean to say, sorry. The efferent arteriole makes a loop. Before leaving the nephron around the loop of Henle. Need to redraw this somehow went away. This efferent arteriole makes a whole networking around the loop of Henle in this way. Okay, and this networking around the loop of Henle is called as what? Is called as, we'll name this also, is called as vasa recta, around the loop of Henle. And this whole networking structure is basically given a different name. It is called as, just name it this side, peritubular capillary. So let's see once whole of the diagram, whole of the thing summed up together. First of all, there's an arteriole which is named as afferent arteriole. A for apple. So this afferent arteriole, as you can see, is entering inside a cup-shaped structure, and that cup-shaped structure is called as what? Is called as loop of Henle. Sorry, Bowman's capsule. Okay. So this is the glomerulus section. The tuft of capillaries. What is glomerulus? So if a question comes to you, what is glomerulus? You would answer it to be what? You would answer it to be a tuft of, tuft means group, tuft of capillaries. So these tuft of capillaries, they accumulate at one place to form a networking called as what? Called as glomerulus. This glomerulus is formed when afferent arteriole enters inside the structure called as Bowman's capsule. So this capsule-like structure This whole capsule-like structure, if you just mark it in marker yellow, this capsule-like structure is basically called as Bowman's capsule. Now moving ahead, what you have is next, Bowman's capsule proceeds ahead to form proximal convoluted tubule. Convoluted means highly looped up. So it's convoluted, proximal means present at the initial end. So just going to mark this in blue. So this particular part is said to be PCT. So it's very convoluted, although I have drawn a very simple diagram, just made it straight and shown to you. This particular structure is called as PCT. Now, after proximal convoluted tubule, you have the next thing called as loop of Henle. Since the filtrate, the particular thing is moving down, see, it is going towards downwards. This is called as what? This is called as descending limb of loop of Henle. So this loop of Henle is going down. Loop of Henle is going down, the green colored structure. So it's called as descending limb of loop of Henle. The descending limb again comes up, this chrome yellow color. The descending limb comes up towards the upper side, it starts to move. So it's called now as ascending limb of what? Loop of Henle. Now, this ascending limb of loop of Henle is now going to start a new structure called as distal convoluted. Distal means something which is present at the end. So proximal means at the beginning, distal means at the end. So this is called as distal convoluted tubule. And finally, distal convoluted tubule terminates in a common aperture, in a common tube-like structure. And the common tube-like structure is called as what? It is called as collecting duct. This is a common tube-like structure. It is called as collecting duct. So this is the sum up of whole structure, and this efferent, E for elephant, this is the efferent arteriole. Let's see this section also. Yes, this is the efferent arteriole. When it starts to come out from the glomerulus, it forms efferent arteriole. This goes and makes a round around the loop of Henle, DCT, everything, and this loop is called as peritubular capillary. Now, peritubular capillary in the region of loop of Henle is called as vasa recta. Clear? Very colorful diagram. Clear, children? Clear with everybody? Yes, Shillong? If it is clear, just give me a thumbs up. Yes. Everybody clear with this? Very good. Shall I move ahead? Shall I move ahead? Okay. So let's write about something, write something about them. Okay. So first thing is what? First thing is we will write about something about nephron. So what is nephron? Nephron is the structural and functional unit of kidney. Okay? So this tube is how much? This happens to be somewhat 3 centimeter long, and somewhat 20 to 60 micrometer in diameter. You are aware about the unit micrometer, everybody? 10 to the minus 6 is micrometer. Everybody is aware about it? It's this much in its diameter. So we will divide nephron into following parts. Number 1 we have is Bowman's capsule. So what is Bowman's capsule? It's a cup-shaped structure having double-walled configuration. So it's a cup-shaped structure having a double-walled configuration. Okay. So in the Bowman's capsule, what we have is, we have the second thing, that is the glomerulus. So inside the Bowman's capsule, we have a tuft of capillaries. I told you tuft means what? Tuft means bunch of capillaries called as glomerulus. So what is glomerulus? If a question comes to you, you have to answer in this way, that yes, what is glomerulus? Glomerulus is the tuft of capillaries present inside the Bowman's capsule. Okay. So next question is, next part is what? The afferent and the efferent arterioles. So what is afferent arteriole? What is efferent? Afferent is something which is going into the capillary or the arteriole going into the glomerulus. And what is efferent? It is coming out of the glomerulus. So once it comes out, you have to call it to be efferent. So remember, in alphabetical order, A comes later, A comes first and E comes later. So in that order you can learn that A is the one which is entering. I have learned in this way. A is the one which is entering, so it is A for apple, first is entering and this is leaving the section. So efferent arteriole, E comes later in the alphabetical order, so it is E. So A moves in, A is going into, and E moves out. A, E, in that way you can learn it. Okay, clear? Okay, fine. Next thing we have is the PCT. What is PCT? They are the proximal convoluted tubule. Bowman's capsule continues into it. Continues to form PCT, and this PCT is lined by simple cuboidal brush border epithelium. Just keep this thing on hold. I will be explaining you this thing in today's class itself. Just keep this yellow part on hold. Just write it over there. I'll be explaining you within that 15 to 20 minutes. What is it? So this is Bowman's capsule continues to form PCT, and PCT is lined by what? PCT is lined by simple cuboidal brush border epithelium. Just 20 minutes. Give me 20 minutes. I will be explaining you what is simple cuboidal brush border epithelium. First, let us complete with this. Then PCT continues to form what? It continues to form loop of Henle, which is further divided into two parts. Number one, we have is descending, since it is going down into the medulla, descending limb of loop of Henle, and we have is ascending limb as well. So two limbs of loop of Henle, the fifth part. So following this loop of Henle, what we have is, we have the DCT. Okay. So DCT is the part where ascending limb continues to. So this DCT is distal convoluted tubule, and this has what? This is connected to the next part, that is number 7, called as collecting duct. So this collecting duct, what does it do is, it collects the urine and passes it to what? Passes it out of the medullary pyramid. So what is the part outside the medullary pyramid? We have is into the renal calyx. And then we have is renal pelvis. From pelvis is go down into ureter. From ureter we have the access to urinary bladder, and from urinary bladder it goes out through urethra. So this is the whole pathway of urine flow. How does it happen? The whole things. Okay. So in the meantime, we are left with one more section, that is number 8. Okay, did you copy this? Sorry. Everybody copy it? Children? Clear? Copied and clear? Yes. So this section is divided into two parts actually. You have, just I will draw a line. This is already very colored diagram. Let's take a marker to draw something. Okay, in purple I can use, since we have not used purple anywhere. So obviously we will divide it into two sections. The outer is called as the cortex. We did this in last class also, I guess you must be remembering. And the things below the purple line is said to be the medulla. So we have two parts. One is cortex and other is medulla. Okay. So cortex is the outer part and medulla happens to be the inner part. Clear? Okay. Now we have something else also. I told you, telling you something else. So we'll talk about peritubular capillary. So what is peritubular capillary? This is efferent arteriole. What does it do? Loops around loop of Henle in form of network. called as what? Next is vasa recta. What is this? This is peritubular capillary in the region of, which region? In the region of loop of Henle. Yes, Testo College, how are you? Good evening. How are you? Good? Yes, children? How are you both? How are both of you? Okay. So everybody copied? Yes, children. Did we copy everything? Okay. Nice. Okay, moving ahead. Okay, so let's see the structure of Bowman's capsule first. Very bad, I do not like it. This is a particular structure of Bowman's capsule, and this is what? This is the endothelium of, just like in alveoli we did, we are going to have the same thing in Bowman's capsule as well. So the endothelium of blood glomerulus is the first structure, and this is the third structure, that is the, so epithelial is any and in between we have is number 3 basement membrane, and in between these structures what do we have is, we have certain foot-like processes, draw in orange better. We have certain foot-like cells, and what are these foot-like cells called? These cells are called as podocytes. Okay. Now what happens is, you have a flow of blood or filter crossing this section and coming down to this area, the flow of filter. So this particular part is called as filtration slit. through which substances are coming down. So you call this section to be what? You call this section, so substances are coming down. So you call this section as filtration slit. Okay? So everything which is present in the blood part, this is the blood part, we know. What is this? This is the blood part. So blood is the part of the blood which is liquid in nature. We have done those sections. So it's called as plasma. So plasma comes down from the blood and forms what? Forms filtrate. So when plasma is coming down from the blood to the filter and gets filtered and enters in the filtration slit in the Bowman's capsule, it is called as filtrate. Clear? Clear with everybody? Yes? No? Tell me something. Yes, Testo College, Shillong. Am I clear up to this? So this is the particular part where blood is flowing towards this section and in this towards this section. Nice? Okay? So this was the first part of the mechanism of urine formation. So we'll start up a topic over here. I was supposedly going to start over here only. So this is the first part of, we have to start up with a new topic, that is mechanism of urine formation. So this is the mechanism of urine formation. Under this mechanism, the first thing what we have is, number one we have is glomerular ultrafiltration. So this is what is a glomerulus? This is ultrafiltrating the things through 3 different types of membranes. Let's see once again. The first is the endothelium of the glomerulus. Second is the basement membrane, and third happens to be the epithelium of the Bowman's capsule. So when the whole processes, the whole membranes are passed through, then only we say that yes, the filtration part is now over. And what is getting filtered? Let's write down something under this heading. So what happens is, it can be ultrafiltration, it can be filtration, both of the things are correct. नहीं तो yes. So we have 3 membranes through which the whole process of filtration is going to take place, isn't it? So what is the end product of the filtration part? If we talk about the Did you get this thing, everybody? Tetso, Shillong. There's those things written in text matter so that you can write in your school exams. Yes. Did you get it, Shillong? Yes. I'll be giving you some questions after this. Just keep a hold on this thing. Clear? Yes, Shillong. Tetso. Shillong can show me a thumbs up if they are clear with it. Yes. Shillong, everybody, give me a thumbs up if it's clear. It's not? Is it? What is not clear? Tell me. Yes, children. What is not clear? Yes, that's so. Are you getting things? You joined a bit late. Are you getting, children, everything? Okay, so let's see what is ultrafiltration product. So what is going to happen? Plasma. This plasma. Now we have studied plasma that it has certain ions. Oh, you're giving thumbs up, but I don't know why I'm not able to see. Okay, that might be because of poor camera reflection. Okay, fine. Thank you so much. So what we have is now plasma gets filtered without the protein part, isn't it? So protein will be remaining in the up section, that is in the glomerulus part, and only the plasma section minus the protein is going to come downwards in the Bowman's capsule. So plasma gets filtered without the protein or plasma minus proteins, isn't it? We know what is the particular proteins present in the plasma. And this gives you what? This gives you about 20% of the plasma forms. We're going to use a new term called as filtrate. Forms filtrate in the Bowman's capsule. Okay, 20% of the plasma will come down. So suppose you are taking off at one round, you are like passing 100 ml of blood through this section. Let's see the diagram. So if you are passing down 100 ml of blood through this section, suppose to this part, out of the 100 ml, you will be having 20 ml in this part. So this part is going to have 20 ml of that particular 100 ml. So it is having what? It is having 20% of the things passing down. Okay? Clear with the idea, everybody? Okay. So this is what is called as the rate of plasma filtration or the rate of filtrate formation in Bowman's capsule is called. So the rate at which the filtrate will be formed is called as what? It's called as glomerular filtration rate. So what is glomerular filtration rate? Is a rate at which the filtrate is formed in the Bowman's capsule. So whatever rate the Bowman's capsule is forming filtrate, it would be called as what? It would be called as GFR. So GFR normally is how much? It is 125 ml in a single minute. Okay, can you calculate the GFR in per day value? Can you calculate it? This is GFR for a single minute. Yes, Shillong, Tetsu, can you calculate the GFR for whole day? Quickly. Yes. No, yes. Can you calculate? GFR in one day, quickly. If it is 125 ml in a single minute, how much is it for one day? How many minutes in one day? How much is it? Yes. How much is it? No idea. Yes. What is taking so long? Quick. Simple unitary method you have to apply. How many minutes in one day? How many minutes in one day? One day equals to how many minutes? It's basic maths. You need to do this much of maths. No, it can't be. Cross check it. It should be 180. Cross check it, please. So one day is how many hours? 24. How many minutes in 24 hours? 60. Isn't it? 2460, how much is it? Tell me. How much is 2460? Basic maths. Yes. How much is it? So if it is 1400, how much? Minutes. So 1 minute equals to how much? 125 ml. So how much would be 1440 minutes? 125 into 1440. How much? Again. Is it this much? See, I have to teach you these things also, huh? Not good. Basic maths. ml per day, would it be? It would be. So if it is 18000 ml per day, can I convert it into 180 liters per day? Yes, I can. So you have what? You have this much amount of filtrate formation every day. So every day your plasma is getting filtered, is coming down in the Bowman's capsule in the value of this 1800 liters. So filtrate formation. So how much urine you are releasing? How much urine you are releasing per day? This is the filtrate formed. Per day, isn't it? But we produce this much urine only. In a single day, you are producing this much urine only. How much is it? It is 1 to 1.5 liters only. So where is this whole 179.5 ml going? Not ml, sorry. This would be liter, not ml. So where is this going? Yes, any answers? So what is the whole story like? You are forming this much of filtrate every day, but we are forming only this much of urine only every day, 1 to 1.5 liters. So my question is, where is this 179.5 liter going? This is reabsorbed by your body. This whole 179.5 ml, liters of filtrate is reabsorbed by your body by a process called as what? By a process called as tubular reabsorption. Okay. So you have a process called as tubular reabsorption, and through this process what happens is you are going to reabsorb the 179.5 liters of urine. I'm sorry, filtrate. So what is tubular reabsorption doing? We will not call it urine. I will tell you the reason why. Or filtrate through different parts of nephron. We will see now the thing what I kept on hold. So through different, exactly, it's absorbed by blood, very good. So through different parts of your nephron, what you are going to do is that you are going to reabsorb all those 179.5 liters of filtrate. So since we are terrestrial animals, we cannot lose this much amount of filtrate from our body because that would be making us dehydrated. This is huge volume. You have only 5 liters of blood in your body and you cannot afford to lose 179 liters in a single day. That is ridiculous, isn't it? So what you are doing is you are repeatedly doing rounds of things, rounds of activities to reabsorb these particular products from the different parts of the nephron. Clear? Sorted? Everybody, let me know. Tetso, Shillong. Tetso, clear? Shillong, very good. Clear with this? Okay. So I will be giving you 2-3 questions. After that, I will be giving you a small break. So first, let's do some questions. Okay. First question. Yes. Just replace this option. It's good. Yes. Start answering. Shillong, Tetsuo. Okay, very good. That's all your answer. Good answer, children. Yes. So this obviously is loop of Henle, because if you see the diagram, just show the diagram, this particular thing. See this thing. So obviously loop of Henle is the only part which is present where? Which is present in the medulla section. Isn't it? Apart from that, everything else in the options are present in the cortex region. Next question. Vasa recta, tubular capillaries around, obviously this would be loop of Henle. Very good. Keep it up. Full marks. Okay. We'll have a small break. Do you want a small break? Want a small break? No, yes? Shall I continue? Okay, I guess you people are also in dispute whether to have or not. Let's have a small break of 5 minutes. Okay. Yes, children, can we resume? Everybody is back? Let's start. Okay. Yes, so in the last section what we did was, before the break, we were talking about a particular process called as tubular reabsorption. Okay, so what is tubular reabsorption? You are going to reabsorb 179.5 liters of filtrate through different parts of nephron. Okay, so now let's see which part of the nephron is going to do what for us. Okay, precisely talking about, let's start up with PCT, because Bowman's capsule, there is no filtration process, there is no reabsorption process taking place in the particular section. We'll start up with PCT. So what is PCT? The second part, isn't it? So if we talk about Bowman's capsule, is there something in Bowman's capsule reabsorbed? No. We'll start up from PCT, and remember with PCT we talked about a particular kind of membrane. Just we'll take you to that particular section. That is what was simple cuboidal brush border epithelium, isn't it? Simple cuboidal brush border epithelium. So what PCT has is this typical structure. So let's see. So what is simple cuboidal brush border, epithelium. Okay, this is simple cuboidal brush border epithelium. This is basement membrane. Over the basement membrane you have some cuboidal cells. This is cuboidal cells. Simple means only in one layer. Just one layer of it. And cuboidal obviously means the shape of cube. Brush border means it has brush over all its surface, somewhat like this. Yes. So what is simple cuboidal brush border epithelium is supposedly going to do? This is a basement membrane. Every epithelium has a basement membrane. Chapter number 7, that is a particular chapter, we are supposedly going to go in detail with these things. But for now, we need to understand this particular part, that every epithelium has a basement membrane, and over the basement membrane, there are different kinds of cells. This is what we are talking about, cuboidal cells with the brush border. So what is this brush border doing? This brush border is actually increasing this whole structure, epithelium, this simple cuboidal brush border epithelium. What it does is that it increases the surface area for reabsorption. So what is supposedly doing? It is increasing the surface area for reabsorption. So you will be reabsorbing more substances through this particular cuboidal brush border epithelium. Okay? Clear? So what we do is that nearly we reabsorb 70 to 80% of the electrolytes, because whatever is in plasma minus the protein is going to be a part of filtrate. Okay, so nearly 70 to 80% of the electrolytes will be reabsorbed, because yes, we cannot afford to lose electrolytes, we cannot afford to lose the many of the important things that are present in the filtrate. So we want to reabsorb 70 to 80% of the filtrate things. Along with that, we will reabsorb all the important nutrients. Okay, let's see some examples of the electrolytes. What are the electrolytes reabsorbed? We have NaCl, that is Na plus Cl minus. We'll be reabsorbing K plus. we will be reabsorbing water, yes, and bicarbonate ion. So we will be reabsorbing these particular things from the electrolyte section, and we will be reabsorbing some important nutrients like glucose, amino acids, and some vitamins. Obviously, talking about water soluble vitamins. There are two vitamins which are water soluble. You can learn it using WBC. W is obviously water soluble. Which vitamins? Vitamin B and C. So we have vitamin B and C, which are what? Which are water soluble. So we have these particular things going to get reabsorbed from the PCT part. This is the PCT section we're talking about. See, the PCT part is going to reabsorb these particular things. Clear? Everybody? We'll draw a diagram after this to see the reabsorption from the different parts of the nephron. Summarize with the diagram. Done, children? Tetsu, Shillong, have you copied this part? Yes, everybody. Can I move ahead? Yes, children. Okay, copy and clear. That's a protocol, is it? Okay, let's draw a stick diagram to have a different view of the different sections. This is a simple stick diagram. You can draw a nephron also, no issues with that. I'll be using this diagram. Okay. So this was glomerular section, this was Bowman's capsule, remember? Just now we did with Bowman's capsule, so there is no reabsorption taking place in the Bowman's capsule. We'll talk about this part. Now, this part is going to release what? First and foremost, talk about HCO3 minus. Then we have NaCl, small amount of K plus, water, what else? Glucose, amino acids, is it? Anything else? Anything left? Just cross check. We have done NaCl, K plus, water, and glucose and amino acids. Okay. Nothing is left. Done with everything. Okay. Clear? This was the region of PCT. Next, proceeding to third part. This was the region of talking about PCT. Wish to draw it? Draw it quickly, please. Yes, vitamins. Okay, I'll add up vitamins here only. Very typical about vitamins. Good. Yes, Shillong, that's so clear with this? Everybody? Are we clear? Okay, nice. Third section, can we move towards third section? That is descending limb of loop of Henle. I will write loop of Henle as LOH. Now on the descending limb only we reabsorbed water. Only water is reabsorbed. Next section is ascending limb. Loop of Henle. Now this is only water is reabsorbed, plus you will add one thing, it is impermeable, that is it will not allow any ions to be reabsorbed. Now this is only ions are reabsorbed. Add up. Impermeable to water. So just reverse, the other way around. First, the descending limb, we don't have any permeability for ions. In the ascending limb, we don't have permeability for water. And what are the ions which are basically reabsorbed? We have NaCl. Okay, clear? Let's see this thing also in the diagram. So you're going to reabsorb what? You're going to reabsorb a huge volume of water from the descending limb of loop of Henle. And from this section, what you are going to do? You are going to reabsorb all the NaCl. Clear? Everybody? Yes, children? Tetso, Shillong, tell me, Tetso, where are people? Let me know if it is clear up to this. Yes. Yes, children, clear? Can I move ahead to DCT now? The DCT part. So what is DCT doing? DCT is also helping in reabsorption, but how? DCT is doing something which is basically your hormonal regulation. We will see this thing also sometime later. For now, you need to see is that there is reabsorption of what? Na plus. Reabsorption of Na plus and so do you remember osmosis part? Did I made that clear in the first section I was teaching you before this class? Did we talk about two solutions having different osmosis? Did we? Did we talk about two sections having different osmosis, children? Tell me. No? Yes. What was it? Okay. We'll tell you now. It's followed by reabsorption of water. Okay? There is also reabsorption of HCO3 minus and Cl minus. So if we talk about DCT, what is the thing going on? Na plus, Cl minus, first of all Na plus, which is followed by H2O. Then we get to have reabsorption of Cl also. We reabsorb Cl from this part. We'll absorb some HCO3 minus. I will write HCO3 over there, that area, for a reason. So this is the reabsorption section of DCT. Clear? Everybody? Done? Okay. This reabsorption of Na plus, let's see this something. takes place under a hormone called as aldosterone. So what is aldosterone? Aldosterone is a hormone from the adrenal cortex. There is a particular gland, okay, there is a particular gland called as adrenal medulla. You must have adrenal gland which is present just above your kidneys, okay. Now from that adrenal cortex, the outer part is secreting a hormone called as aldosterone, and that Na plus reabsorption takes place from that particular, because of that particular hormone called as aldosterone from the DCT section, okay. Now if you take two solutions, this is what I was talking about, osmosis phenomenon. If we talk about two solutions, for example, solution A having, just take an example, suppose 10 molecules of, and so B having suppose 30. Let's talk about millimolar. I guess moles concept has been made clear by Sube sir. Suppose it's 30 millimolar and this is 10 millimolar. Have you done this question? 250 ml of water in each. Have you done this question? What is going to happen? First option. Na plus moves from, this is osmosis I am talking about. The phenomenon of osmosis you have to explain. Na plus moves from A to B. Next is Na plus moves from B to A. D. Water moves from A to B. Sorry, this is C. And D. Water from B to A. Yes. Tell me what is right. Which of these options is correct? Any idea? Osmosis. Sure. Think about it. Are you sure? Very good. Keep it up. So water is going to move from B to A. This is what is called as osmosis. Why B to A? Because here water is in higher concentration. See, there are more free molecules of water in B. And here water is in less free value because this is somewhat more concentration. So if it's more concentration, there would be less free water molecules. So it would need more water from the surrounding, and this is always and always separated by a semi-permeable membrane. Isn't it? So this same thing is happening over here also. When you are reabsorbing sodium, when sodium is being reabsorbed in the plasma, it is followed by reabsorption of water. This is a simple mechanism of osmosis. As water moves from its higher concentration to its lower concentration to maintain equilibrium. Very good. Okay, fine. So that was how we justify the reabsorption of Na plus followed by water. Next thing is ascending DCT. We'll talk about the sixth part, that is collecting duct. What is reabsorbed over here? Let's see. In the collecting duct, there is absorption of, so we can say reabsorption of water under the effect of ADH. So ADH is what? ADH is anti-diuretic hormone. What does it do? It increases the reabsorption of water from filtrate in plasma. Now one more golden rule I'm going to give you. If we talk about the process of reabsorption, suppose I'm talking about the process of reabsorption. You have to always take into account that reabsorption will be always from filtrate, without any exception. Reabsorption is always from the filtrate. You are going to reabsorb things from the filtrate without any exception. And if I am talking about secretion, the next topic will be secretion only. If I am talking about secretion, it would be always what? It would be always into the filtrate. So we have two things. What? We have reabsorption and we have secretion. So reabsorption is always from the filtrate and secretion is always into the filtrate. Am I clear? Yes, children, am I clear? Tell me, Shillong. Tetso. Clear, children? I guess we must be. Okay. So I'm moving ahead. Okay, fine. So what is this happening over here again? We are going to reabsorb huge volume of water under the effect of what? Which hormone? Under the effect of ADH. So done with this? Diagram is clear? Yes, children. This thing is done by you people. You have made this diagram. Okay. Nice. Very good. I'm moving ahead. Okay. So next section comes is the third part of urine formation. That is a C section. What is that? That is happens to be tubular secretion. So what is tubular secretion? What is it? What is tubular secretion basically? This is basically the secretion of substances into filtrate. Okay? This is an active process. Obviously, if you mean active process, this would be requiring ATP. We know these things. This is very basic. So it's an active process which is basically in three parts: PCT, DCT, and collecting duct. Okay. In PCT, what are being reabsorbed? Sorry, what is being secreted? Let's see. We have H+, we have NH3+, we have K+. So K+ is both reabsorbed as well as secreted. I will tell you the reason why. Just wait for 2 minutes. Okay. DCT is going to reabsorb what things? DCT is basically going to reabsorb. Now, apart from this thing, you have some other things also. You have H+, K+, and NH3+. Same things. Okay? Clear? And collecting duct, what is it going to reabsorb? Collecting duct is basically going to reabsorb small amount of H+, and K plus, negligible. In fact, you need not mark in your diagram also. Okay, let's see the diagram once. We'll mark over here also. So we are putting it into H plus. We'll be putting into it NS3 and we'll be putting into it same amount of K plus. Okay, this side we are going to put K plus, NS3 plus, H plus. So wherever you are having HCO3 minus C, wherever you are having HCO3 minus reabsorbed, you have to put into H plus. Next thing, if you are reabsorbing Na plus by active transport, by what transport? By active transport, that is you are using ATP. So if you are reabsorbing Na plus by active transport, you need to put into K plus also. So you have to put K plus if you are reabsorbing Na plus by active transport. Okay, this is sodium potassium pump. You must have heard about it. It's called as sodium potassium pump. What is sodium potassium pump? You are reabsorbing sodium and you are just putting into it potassium. This sodium potassium pump. Same thing is operating here also. See, active transport of sodium out, active transport of potassium in, sodium potassium pump. You are moving out HCO3 minus out, you need to move H plus inside it. Clear? That's why we are putting K plus and we are reabsorbing also K plus in the PCT section. Okay? A small amount of urea is released from this part, which is reabsorbed by the ascending limb of loop of Henle also. Clear? Everybody? Yes, children? Shillong, Tetsu, are we clear with this? Yes, Shillong, give me a thumbs up. Are we clear? Clear? Okay. 2 times clear means very much clear. Okay. Copy it up to this, everybody? Let's start up with composition of urine. So what is urine composed of? First of all, there is 1 to 1.5 liters of urine produced per day. Average, obviously, that differs from like season to season, access to water, all those factors. Color of urine is pale yellow. And this is because of what? This is because of a pigment called as urochrome. Okay? So there is 25 to 30 grams of urea. Since we are ureotelic, so we excrete out 25 to 30 grams of urea per day. The pH of urine 6.0. So we can call it to be slightly acidic. Happens to be slightly acidic in nature. Clear? Done? Shillong? So we are having 4 topics more. First we have osmoregulation. Then we have is regulation of kidney function, accessory organs and disorders. Okay. So what is going on in your school? Shillong, what are the topics you are going ahead in your school in biology? Yes. What is being taught in your school now recently, or whatever has been completed? Tell me. Okay, you are from different schools. So is this chapter being done in your school? Has this chapter been done in your school? No, I'm just asking you this in concern why, because what chapter should I start after this? That is what is my question is next. Next I'm going to follow this question. What am I supposedly going to start after this chapter? Shall I move to animal kingdom? Or should I continue with the human physiology? Yes. What do you wish? Shall I continue with locomotion and movement after this? Or should I move to animal kingdom? Or biomolecules? Yes, we can start biomolecules. Okay, fine. So after this chapter you wish to start biomolecules, is it? Okay, no issues. We can. Okay, fine. We'll start up with biomolecules after this. No issues. Okay, fine. So let's have a quick revision and let me know if you have any doubts up till now in this class. So we'll start up with the things. So we started with a short revision of the topics in the last class, and then I taught you about the structure of the nephron. Very colorful, very super, in fact, hodgepodge. Okay, then we talked about nephron structure, and I gave you all data. So see, these data are not important. You do not learn these things. This is just to have a particular thing in your mind. Okay, this is this long, this must be this wide, only that thing. This is just wow factor. Okay, don't learn these data. This is useless. I never learned it. Okay, so no need to learn these data. That is, this is 3 centimeter long. Some might be telling it 3.2 centimeter long. You will be then it will be fixed. So just go ahead with this thing. Just wow factor. That oh, this is this long and this is this much. So how many nephrons are present in one kidney? Both of your kidneys contain like 1 million nephrons together. Huge number. So they must be super complex in their structure. This is what is a simple diagram we have drawn. Just made it simplest way possible. Although it's super hodgepodge, I agree. Then see 3 centimeter long. After this, we have divided into different parts. I told you every important detail about the parts, and this is somewhat very, very important if you are appearing for your NEET or as well as your school exams, because they do ask this for 1 mark. This is a 1 mark question. This can be asked for a 1 mark, for a 1 mark question, that yes, tell us the particular activities, type of cells in the simple cuboidal brush border epithelium. And we talked about the same thing was going on. I told you about peritubular capillary. It is an efferent arteriole making a loop around the loop of Henle. Okay? Mechanism we started. Ultrafiltration was the first mechanism of urine formation. And we saw how filtrate was formed through passing by 3 membranes. Okay, 20% of the plasma is going to form the filtrate with a rate of 180 liters per day. Huge. We are reabsorbing 179.5 liters by tubular reabsorption. Okay, this reabsorption process is very high in PCT. So you are reabsorbing everything in PCT, in fact. And if you talk about reabsorption, tubular reabsorption, we need to mention a point that 99% of the filtrate is reabsorbed. Just mention this point also. You are going to reabsorb 99% of the filtrate. 99% of the filtrate is going to be reabsorbed. Then we have basement membrane, cuboidal epithelium, and we place cilia over them to increase the surface area, and we call this structure simple cuboidal brush border epithelium. This was the whole section. I hope so you have used multicolored pens to show the secretion and reabsorption section. If not, go back home and just use a multicolored pen. A different kind of pen for reabsorption and secretion. That will make the diagram very, very easy to understand. This urea section I will be covering to the next class. Why this urea is being released and then reabsorbed. First it has been reabsorbed and then again secreted. We'll cover this section in the next class. What is happening over here? The whole suspense. Then we have descending, ascending, the roles played by them, the aldosterone factor. Then we talked about osmosis, because we talked about followed by. So I told you the mechanism of osmosis. You answered it correctly. Very good. Then we have collecting duct. From there we have reabsorption, secretion, tubular secretion of substances into the filtrate. Yes, it was an active process. ATP is required. What are the things? Almost similar kind of things are being reabsorbed too. And finally we landed with the composition of urine. Okay. I'm left with micturition. The next class will start up with micturition. Let me see. A small part of this is left, micturition. We have osmoregulation and regulation of kidney function. Let's see if we can complete. We'll start off with biomolecules in the same class. So we have other excretory organs also. And we are left with disorders. So 1, 2, 3, 4, 5. 5 topics are left. If we can finish it that day, we will start with biomolecules same day. Just go back home and do some questions. Try to answer. Okay. One thing more. Your NCERT, your books, whatever books you are following at your school, your state board, or you are following your NCERT, whatever book you are following, just go back home and try to do some questions out of this. Apart from that, I will be giving you 5 questions in assignment. You have to go get those questions done in the next class. Okay? So first question is, just write quickly. Let's see, we'll do 3 only today. Composition of urine, components that are reabsorbed in different parts of nephron. We'll write reabsorbed and secreted also, same question. And third and last question we have is well-labeled diagram. Okay, draw a well-labeled diagram. Showing different parts of nephron. So these 3 questions you have to get them done before you come to the next class. Okay, this is your homework. You do it and come in the next class. Okay, children, do you have any doubts? Any problem, any issues? No, no problem? Shillong, Tetsu was very quiet today. I don't know why they came late. I guess they are connected. But there's some issue, I guess. Okay, fine. Yes, children, any problem? Shillong. Shillong. Yes. Any problem? No? Everything is clear? Okay, then I am expecting answers, many answers on the group. Just draw those questions, do those questions, draw the diagrams and send them on the groups. Okay? Fine? Okay, fine. Okay then. Thank you so much. Do your homework. We'll meet in the next class. We'll try to complete this chapter. Obviously, we're doing some questions. And we'll start with biomolecules, if possible, the same day. Okay, bye-bye.